Okay, this is our third review, and it is on cellular energetics. I'm just going to put up the PowerPoint here. All right, so review session 3. The topics we're covering in this unit, we're going to look at enzyme structure, how they work, and then the environmental impacts, which we did in the enzyme lab. It's your first formal lab for me. And so you can go back and look at that. You can also, again, do the lab bench on Page School if you want to look at that to see how else it could be done. Then we're doing cellular energy. That's about ATP and coupled reactions, photosynthesis. And we did a lab on that. If you remember, we did the leaf chat lab. So we looked at different environmental factors that affected photosynthesis, and then cell respiration we also did a lab on, and that was the different types of seeds, or germinating versus non-germinating, or temperature. And then the last part that is in the curriculum guide is fitness. Right. So the part 1, this is on enzymes and cellular energy. So this would be chapter 6 in your book. Okay, so you're looking at biological concepts and/or processes. So you need to understand the science behind it, the identifying experimental procedures that are aligned to a question and appropriate controls. And then also you need to be able to justify those controls. So why does this make a good control or not? And then again, providing reasoning. So we're seeing those science practices throughout the whole curriculum. So remember, if you're getting a question on the AP exam, it's going to be a multiple choice that will contain content and a science practice, or you could have an essay with both content component and a science practice component. So you have to integrate those two. So enzymes are organic catalysts, and we talk about them as though they're specifically proteins, but we have found out that RNA can also act as an enzyme, and that's part of the RNA world hypothesis, evidence that the first genetic material was RNA-based, not DNA. But enzymes have an active site that is substrate specific. So it is going to bind to the active site here, right? And it has to fit that active site. There is an exception to this that we study in photosynthesis, and that's Rubisco. So if you remember, Rubisco, if there's a lot of oxygen, it can pick up oxygen instead of carbon dioxide, because when it evolved, having oxygen in the atmosphere was not an issue. But the problem is if the plant picks up oxygen in the Calvin cycle, instead of carbon dioxide, instead of being a reducing reaction, it becomes an oxidizing reaction, and you're not going to make anything useful. You're going to just use up your ATP, your NADPH, and you're not going to make the glyceraldehyde 3-phosphates. You're not fixing carbon dioxide. And the plants that deal with this effectively are your C4 plants. This is the plants that had this problem, so they adapted, and they are going to have their photosynthesis, Calvin cycle specifically, occurring in the bundle sheath cells. So carbon dioxide gets put into those bundle sheath cells, so you always have a high concentration. It does cost them energy. That's still why most plants are C3 plants, because they're not dealing with this photorespiration. But I wanted to give you that there is that exception. But for the most part, enzymes are going to be substrate specific. How they work is the induced fit model. So the enzyme is going to be basically, it's like a handshake. That's what we usually use. So they come together, and they have to be compatible with the shape and charge at the active site, and then they wrap around each other. Okay. So I'm going to show you a quick video on it, just so that you can help visualize it. There we go. So you can see the enzyme in red, and then you see the substrate in blue, and you see the enzyme wraps around that substrate, and that's your induced fit. How do enzymes work? Well, they're going to lower the energy of activation. So if you remember, this here is an exergonic reaction. It's releasing energy. So we see that there's more energy in the reactants than the products, right? And so our delta G, or Gibbs free energy, the energy available to do work, is negative delta G. That doesn't change whether an enzyme is present or not. What does change is the amount of energy needed for activation, or sometimes they say activation energy. You see it gets lowered. That's the energy needed to get the reactants to the transitional state where the bonds are no longer stable. Okay. So the enzymes are going to lower that. And there's a nice diagram in your book. I just want to give you the page 126 at the top of the page that shows the difference between an uncatalyzed reaction and a catalyzed reaction. There are several environmental impacts on the enzyme function. We studied a number of them when we did our enzyme lab. So I'm going to start with the ones that can cause an enzyme to denature. So this is disrupting the bonds so that it loses its structure. Remember, proteins in order to function need to at least be at tertiary structure. Many will be at quaternary. But if I have these different factors, I'm going to interrupt these bonds, and that's going to cause it to lose that 3D shape. So if it comes down to, for instance, primary shape, just made of peptide bonds, that has no 3D structure, so it's not going to be able to act. So we're going to look at first temperature, pH, and then salinity. You should be aware also can cause this, but we're not going to look at that now. So temperature, below optimal temperature, it is not going to be due to denaturing. It's actually due to the kinetic energy of the molecules. They're too slow, so it's unlikely that this is going to bump into the substrate of the enzyme's active, or the substrate is going to bind into the active site of the enzyme. Okay, so they're moving very slowly. They're not bumping into each other. And that's, you can see on the graph on page 129, the top graph, figure 6.16 A in your book. Okay, as it's moving toward optimal temperature, you notice that the enzyme's reaction rate is going to go faster, and that's because the molecules are moving faster. They have more kinetic energy, so they are bumping into the active site more frequently. However, above that optimal temperature, the molecules are moving into the enzyme so fast that they're literally tearing it apart. They're just bombarding that poor enzyme. So it's losing its structure. It's getting denatured. So just keep in mind, below optimal temperature, it really has to do with the kinetic energy of the molecules and that they're too slow to bump into the active site. But above optimal temperature, it's about denaturing. Right? Sometimes students put denaturing with below optimal temperature, and that's a mistake. And that's what we see here. There's a nice explanation on the side, and it explains that denature. So here's our optimal temperature. Now remember, this one shows 37 degrees Celsius, but that's not going to be true for all organisms. You have the archaea bacteria, the thermophils, that we get Taq polymerase from. They're working at a much higher temperature. Your dog's temperature is around 102 degrees normally. So it's going to have enzymes that work at a higher temperature than ours. All right. How pH works on enzymes. Enzymes also have an optimal pH. And in that same figure on page 129, you will see the B1 is all about optimal pH. So I'm going to stick with that kind of classic example. We have pepsin here. That is going to break down proteins, as is trypsin. But they're working in different compartments of the digestive system. So you may remember when we studied the digestive system, we talked about that compartmentalization of where things got digested. And it was because the enzymes are working at their optimal pH. Okay, so pepsin works at optimal pH around 2. So it needs to release a hydrochloric acid to make that pepsinogen active. But when that acid chyme enters the small intestine, the trypsin, still breaking down proteins, is only going to work at an optimal pH of 8. So the pancreas had to add bicarbonate in to up the pH. That helps denature the pepsin and make sure the trypsin and the other enzymes working in the small intestine are the only ones that are functioning. Below or above the optimal pH, hydrogen ions and hydroxyl ions disrupt those hydrogen bonds that provide the enzyme its structure. So again, you lose structure, you lose function, because structure defines function. For concentration of substrates, the reaction rate will increase until every active site is filled. That's our saturation point. And that's actually your rate of reaction as well. So I could easily see them having you graph some of this data and then having to explain it, identifying optimal pH, optimal temperature, or reaction rate. So you should remember with reaction rate, it's where it levels off. And that's because you're waiting for that active site to be open again in order to turn the reactants into the products, right? So you have to wait for one whole reaction to occur. So here we see the substrate again, but in enzyme concentration, we're going to have this exponential kind of growth here, as long as there's substrates available. Regulation enzymes, this is usually going to be negative feedback. So remember, negative feedback is like a light switch that turns something on or off. So I have enough of the end product. This could be in a catabolic reaction, I've broken something down, or an anabolic, I've built enough of this up, and that goes back and it turns off the enzyme here at the beginning. And so the reason for that is so that you don't waste energy and other resources building or breaking down something that you don't need, you have sufficient of. Right? So the way it can be regulated, one way is competitive inhibition. So that's going to mimic the substrate, and it can bind reversibly or irreversibly to that active site. Right? So it's binding here to the active site, and now my substrate can't bind. Sometimes it can be overcome with more substrate. But an example where it can't is cyanide, the poisoning. That binds to cytochrome in the electron transport chain. And so now that cytochrome, which is part of the electron transport chain, can't grab those electrons. And so you can't create an electrical chemical gradient to make ATP. And therefore, you die. Non-competitive inhibitors, these bind to a site other than the active site. They change the active site so that the substrate can no longer bind. So it's actually changing that structure by binding chemically to somewhere other than the active site. You have a nice diagram of this. Again, in your book, this is on page 130, figure 6.17. Allosteric regulation, you also have in your book. It's on page 131, figure 6.18. So here you have a site other than the active site, but that regulatory site is either going to have an inhibitor or an activator. So you're constantly turning this enzyme on or off. And my favorite example, as you know, is phosphofructokinase. This is the one that adds the second phosphate to the fructose. So you get a 1,6-biphospho fructose, all right, and that's going to really make that fructose unstable. So it's going to be turned on by adenosine monophosphates. Remember, that's what happens if ATP has lost 2 phosphates. It really is a signal that you are way too low on energy. So that's going to bind to the phosphofructokinase as an activator and allow more glycolysis or allow glycolysis to occur. Right, which you need to happen in order to make more energy. However, if you have an excess of ATP or you have citrate, if you remember, that's the first part of the Krebs cycle. So you have that acetyl-CoA coming in, it's going to bind to oxaloacetate to form citrate, right? So if I have a backup in the Krebs cycle, I'm going to have an excess of citrate. So that will bind here, and that's going to turn off. It'll be an inhibitor for phosphofructokinase. I should mention at this point, you do not need to know all the different parts of the citric acid cycle. We will go over what you need to know in the future. Okay. Enzymes, they're in metabolic pathways. And so what we'll find is the end product of one reaction becomes the reactant in the next reaction. Right? And in order to do that, enzymes are often compartmentalized. They can be in organelles. They can be built into membranes. And so that keeps the reactions going. In genetic metabolic disorders, sometimes one of these enzymes is missing. And so it's going to cause a buildup of this this beginning reactant here. And we're not going to make this end product. Right? So those metabolic genetic disorders, that's the problem. Some can be treated with diet, others medicine, but there's a number of them. Cellular energetics, we know life is ordered. And in order to have order, you have to constantly put in energy. And so if you do not have energy, you're going to lose your order, and that's going to result in death. So not to break our laws of thermodynamics, though, we have to lose most of our energy as heat. So we have these coupled reactions. So if I break something down, that's catabolic. And some of the energy from that is usually used to build something anabolic. But again, most is lost as heat. Or an exergonic reaction is going to break something down. And then we use that energy to run an endergonic reaction. Okay. So oxidation versus reduction, again, these coupled reactions, we just, at this point, you can view them as different names. Chemistry will clarify it a little bit better. So oxidation, you're losing electrons of energy. Remember Leo, okay? Lose electrons, sorry. That means to break a bond. And so you're losing energy, okay? Reduction is gaining electrons, or you're gaining energy. So I lose energy in one thing, I break a bond, most is lost as heat, but some of it is given to reduce in another reaction. So they're coupled. The other thing to remember is something is being reduced, it's acting as an oxidizing agent. If something's being oxidized, it's acting as a reducing agent. So this is an example question from the publicly released 2013 exam. Okay, this is a calculation one. With the current exam, they could take something like this and make it a multiple choice, or they can make a part of an essay or one of the smaller essays possibly. But you can give that a try and see if you can do that calculation and read that graph. That's a good science practice skill. This is an example essay. So you can see here, you would have to, this is on enzymes. You would have to do a graph. You have to do a calculation. So again, science practice skills. You have to do an explanation. So you have to explain the process, right? You have to draw, label another line. You're making a prediction there. You have to explain why you made that prediction in scientific terms. And then you have to do some identification and explain those as well, discuss that. Remember, you want to focus on those task words. I gave those to you, but if you lost that, it's also in the curriculum exam description, which is publicly available for you. All right, our next part is cell respiration and photosynthesis. So if you need to take a break, this would be a good part to take a break and come back. Okay, the last was the cell respiration lab. That was with the seeds. So germinating versus non-germinating, type of seed, temperature. Those were all different variables that you could have looked at. Photosynthesis, that was with the leaf chads. So if you remember, you could have looked at the wavelength of light, distance from the light, the amount of sodium bicarbonate. And so when it was doing the light reactions, the leaf chad, if it got, or the type of leaf actually, the oxygen caused it to rise up. And so the quicker it rose up, more photosynthesis was occurring. And then we did through lab bench, the photosynthesis with the DPIP. Okay, so if you want to review, the cell respiration one is on lab bench, as is the photosynthesis with the DPIP. This one has a lot of science practices that directly relate to those labs. Now, you might not get the exact same lab, so it's important to understand the whole process as well. So you need to support a claim of evidence, biological principles, concepts, processes, and or data. Remember, you must cite specific numbers and data, and please do not forget to give your units. A lot of people forget to give their units. Construct a graph. That means that you need to label your X and Y axis correctly. You need to have it well scaled. You need to include your units. Please include a title with independent and dependent variable. And then your graphing of it is usually a point as well. And provide reasoning to justify a claim by connecting evidence to biological theory. So here you're taking the science that we've learned and you're connecting that also with the data from the graph, right? You need to cite specific data. You need to put numbers down. All right. So cell respiration, we're going to focus first on glucose being converted to usable energy, ATP. So ATP releases about 7.3 kilocalories of energy. It has about 15 kilocalories of energy, so the remainder is lost as heat, so about 7.3 is usable. That is enough to run a reaction without generating too much heat, but ATP is very unstable, so you can't store it. So all organisms have to do some form of cell respiration, either aerobic or anaerobic, even your producers, because even though they make ATP in light reactions, they can't store it, so they turn it into starch, for instance. And then when they need it, they can break the starch back down and make ATP out of it. Okay, so our purpose of cell respiration is to get the energy from those organic macromolecules, specifically your carbohydrates, your lipids, and your proteins. So carbohydrates, remember, have about 4 kilocalories per gram of energy, as do proteins. Lipids have about 9 kilocalories per gram of energy. Right. So what we're going to do is in cell respiration, we're going to take ADP, we're going to take the energy made in cell respiration to make it into ATP. This is a high energy bond, right? And we see that functional group phosphate. It's high energy because these phosphates are negatively charged. They're repelling each other. So you need to have a fairly good amount of energy to hold them together, right? Then when I need that energy, I'm going to break down that ATP back to ADP, releasing that phosphate or transferring that phosphate, and now I can do some work. I can synthesize, make something, right? And that phosphate is inorganic. You're not seeing any carbon attached there, right? So keep in mind, all life has to do some form of cell respiration or fermentation to have usable energy. You have substrate level phosphorylation. That's where the energy for turning ADP into ATP comes directly from bonds, either the breaking of bonds or going from a high energy bond to a low energy bond. And that's what occurs in our first two steps, glycolysis and the Krebs cycle, right? And these are coordinated by enzymes, and that becomes very important, right? So glycolysis, this occurs in the cytoplasm. You don't need to know all the steps. You have to have a general idea of what goes on. So here we have our glucose molecule. 2 ATP are released, and we get 2 ADP. That's going to create that 1,6-biphosphate fructose. Because this glucose is relatively stable, we want to make it unstable. And so those phosphates are on either side of the fructose, and they're repelling each other, and it pulls it apart to make glyceraldehyde 3-phosphate, or G3P. Sometimes you'll see it written as PGAL, which is the same thing, phosphoglyceraldehyde. It goes from that high energy conformation to a lower energy conformation in pyruvate that produces 2 NADHs, a net of 2 ATP. So it makes a total of 4, but we have to subtract the amount that we initially invest. So that's why we say a net of 2 ATP and 2 pyruvate. You'll notice we have lost no carbon at this point. So glycolysis, we see the fermentation in animals. I don't know why this says plants. This is not mine. So it occurs in animals. It occurs in bacteria. It occurs in the cytosol. Right? It keeps glycolysis going because what we're doing here in fermentation is we're going to regenerate the NAD+. We're going to take the NADH that was made in glycolysis, and in fermentation, we're going to oxidize it back to NAD+, so that glycolysis continue to run. No oxygen's needed. And what we're going to create as end products is alcohol or lactic acid as our waste products. Respiration, you're going to break down the carbon the rest of the way. We're going to completely oxidize it. Remember, when that carbon is broken down, it becomes carbon dioxide. We're going to need oxygen. This is going to occur in the mitochondria. It can occur in prokaryotes, and I'll have a little slide on that in a second. And the oxygen is your final electron acceptor because it's highly electronegative. The electrons are attracted to it, and it's going to form water. Our products are carbon dioxide, so all the carbon from the original glucose is going to be broken down as carbon dioxide. We're going to have some water up to 38 ATP. It's going to typically be less than that. And then the NADH and FADH2, but those are going to be oxidized and turned back into NAD plus and FAD plus. Okay, so alcohol fermentation, the pyruvate is going to become ethanol and carbon dioxide. And that's happening in bacteria and yeast. It's used by us in things like brewing, winemaking, baking. That's why you get those bubbles that help rise the bread. But the alcohol itself is burnt off, so you don't need to worry about that. And lactic acid, the pyruvate is broken down to lactate. This happens in fungi, bacteria, human muscles. It's used to make cheese, yogurt, acetone, and methanol. And the purpose is really to take the NADH and to oxidize it so that you have NAD plus available to run glycolysis. The pictures of these are on page 150, figure 7.16, if you want to look at this in your book. Right? So glycolysis in the cytosol, that's going to be substrate level. Okay. Krebs is going to happen in the mitochondria, right? That's going to require oxygen, but it's also substrate level. Electron transport chain is also in the mitochondria, but these are both oxidative phosphorylation, the electron transport chain with chemoosmosis. Fermentation is anaerobic without oxygen. So if it's alcohol fermentation, you're going to get ethanol, which is a 2-carbon molecule, and a carbon dioxide. And you'll get lactic acid, which is a 3-carbon molecule. Okay. So going into the mitochondria, the pyruvate is going to lose a carbon as carbon dioxide. It becomes a 2-carbon molecule called acetyl. The energy from that breaking the bond is used to reduce NAD plus to NADH. And then coenzyme A is attached to this. So that acetyl-CoA is made going into the mitochondria. We kind of simplified it here with one transport protein. Okay, our mitochondria, once it is in here, remember this used to be a prokaryote and it was engulfed in the endosymbiosis. Hypothesis explains that. You have that smooth outer membrane. Then you have these cristae from the inner membrane, that folding. So that folding allows more ATP to be produced because you get a higher surface area. You have this intermembrane space, compartmentalization, plus the electron transport chain has a slightly higher pH, if I remember correctly, than the matrix. So again, compartmentalization. And then we have our matrix. This is where the Krebs cycle will take place. Right. So your Krebs cycle, you do not need to know all the intermediates of the Krebs cycle. It's going to occur in the matrix of the mitochondria. It's substrate level phosphorylation. Okay. So the energy is coming from the breaking of bonds or rearranging from high energy bonds to low energy. What goes in is the acetyl-CoA. It's going to be completely oxidized, meaning we're going to it's going to lose all its energy. The electrons is going to come off. It's going to become carbon dioxide. And so you get 2 carbon dioxides per acetyl-CoA. You would, of course, double this for glucose. You get 3 NADHs, an FADH2, and an ATP. I want to point out, because this has shown up on the AP exam a couple times, the formation of this ATP is indirect. So first you have a GDP becoming a GTP, and that GTP goes back to making a GDP. The energy that's released from that phosphate is used to turn an ADP into an ATP. You can see that on page 143 in your book, figure 7.11. Keep in mind, you do not need to know all those intermediate steps. They're not going to question you on that. Right? So the electrons extracted in glycolysis in the Krebs cycle will be transferred by NADH and FADH2 to electron transport chain. Okay, NADH is made in glycolysis in Krebs. It has about enough energy to make 3 ATP, and we have a total of 10 NADH per glucose molecule. it's going to enter the electron transport chain earlier than your FADH2. Your FADH2 is only made in Krebs. You make, and it has enough energy to make about 2 ATP. 2 FADH2 are made in total, and that's going to enter the electron transport chain after the first proton pump. Right. So you don't need to know all the intermediates in your electron transport chain, but you do have to have a general idea of what's going on here. So when we look at oxidative phosphorylation, we're actually looking at 2 parts. We're looking at the electron transport chain, and then we're looking at chemoosmosis with ATP synthase, right? So we're going to go through the parts of the electron transport chain, and then we're going to go over the ATP synthase. If you want to follow along with your book, again, there's a nice picture of ATP synthase on page 145, but the picture that would probably help you out the most is on page 146, figure 7.14. Okay. In order to have an electrical chemical gradient, remember you have to have an intact membrane so you can create an electrical chemical gradient. You're not having things leak through. You have to have energy to create that gradient. So we're going to have proton pumps, right? And the energy for this to run these pumps in cell respiration is coming from NADH or FADH2, those electron carriers. Photosynthesis is the energy from light that was absorbed by the chlorophyll and the accessory pigments. ATP synthase you need because it's going to have a way to harness that potential energy by the hydrogen flow to reduce ADP to ATP. So our electron transport chain, the energy is provided from the oxidation of both NADH and FADH2. Okay, this is in the inner membrane, and it's going to also use that intermembrane space. And we're going to have proton pumps. There's 3 total. The electrons are going to get passed down this electron transport chain because they are attracted to the oxygen, which is highly electronegative, that forms water. And as they're being passed down, they're giving off energy to run those proton pumps. And those proton pumps are pumping about 2 hydrogen into that intermembrane space, or 2 protons. So our steps, just to go over, NADH or FADH2 are oxidized, so they're losing energy. Their electrons now are passed onto the electron transport chain. The hydrogen goes right into the matrix there, right? As those move down the electron transport chain toward the highly electronegative oxygen, is going to pump hydrogen ion from that energy into the intermembrane space. That creates that electrical chemical gradient. And the final electron acceptor is oxygen to form water. Okay. So that electrical chemical gradient, it has lots of potential energy. Okay? We have lots of hydrogen here, very little hydrogen here. Okay? If I go back, I just want to use this. Oops, let me look for my picture here. Here you saw the NADH come in. It gets oxidized. The electrons are here. The hydrogen's released here. And then it's used, that energy, to pump 2 hydrogen here, 2 hydrogen here, 2 hydrogen here. Lots of hydrogen on this side, concentration gradient. Low here. All of these hydrogen also are positively charged, so they can't go through the phospholipid bilayer. They need a transport protein. So in order to go through the phospholipid bilayer, they're going to have a transport protein, and that's going to be ATP synthase. At the end, the electrons are picked up by oxygen to make water. Okay? So the next part is going through that ATP synthase. When they pass through it, they're releasing their potential energy, both charge and concentration has created. Right? Electrical chemical gradient. The ATP synthase spins. That energy from that is to convert ADP into ATP. Again, nice picture of this on page 145, figure 7.13. Right? This just kind of goes over it. This is from a different teacher, but I wanted to give you another way of looking at it other than just the way that I present it. Okay, endothermic organisms, you can decouple the oxidative phosphorylation from the electron transport chain that generates heat. All right, because endotherms regulate their own body temperature. They are going to need to generate heat. How the electron transport chain occurs in prokaryotes, and they don't have a mitochondria, but instead they're going to use it across that plasma membrane here that we're seeing. And remember, your bacteria that you think of, that has a peptoglycan cell wall, so a protein sugar-based cell wall. Okay. Anabolic reactions in cell respiration. So if I need to build things, I can take things from cell respiration to do that, right? So I can take pyruvate to make glucose. From the acetyl-CoA, every two carbons in that, I could add those together to make fatty acid chains. They're going to be longer than 2 carbons. I could take intermediates from the Krebs cycle to make some of our 20 amino acids. And we can also, whoops, did I just, yeah. All right, and so we can see all of these different molecules going in and what they can make for the organism. Okay, that biosynthesis. I don't know if that's in your book though. Catabolic, we focused on glucose, but you can have other carbohydrates go in. They would start with glycolysis. Lipids and fats, they skip the energy investment phase of glycolysis. So glycerol goes in as glyceraldehyde 3-phosphate. And then every 2 carbons from a fatty acid can go in as acetyl-CoA. Proteins, they do have to lose their amino group, but they can go in as pyruvate, acetyl-CoA, or molecules in the Krebs cycle. So these can also be broken down to make ATP, not just glucose. Right, our next part is photosynthesis. So again, if you need to take a break, this might be a good spot for you to take a break. So photosynthesis, we're going to capture light energy to produce sugars. It first evolved with prokaryotes, which are related to the living extant cyanobacteria, and it's adding oxygen to the Earth's atmosphere. So it's changed it from a reducing to an oxidizing atmosphere. And the evidence from this, from the endosymbiosis, remember circular DNA, we have a double membrane, we have small ribosomes like eukaryote. They reproduce on their own by binary fission. They can produce some of their own proteins. So it's important that you remember that endosymbiotic hypothesis. All right, you have 2 parts of photosynthesis. You have your light reactions. Those are turning light energy into the chemical energy of ATP and NADPH. Don't get this confused with NADH. So think P for photosynthesis. And that's going to occur in these thylakoids. Again, high surface area, low volume. A group of thylakoids are called grana. And then the Calvin cycle fixed carbon dioxide into G3P using the energy of ATP and NADPH that were produced in light reactions. That's the stroma. That's all of this out here. Right? And you see that double membrane in this picture as well. So our sites of photosynthesis, those are going to be your mesophyll cells, mainly found in leaf cells, but any green part of a plant can do photosynthesis. You have your stomata or stoma, it's just whether it's singular or plural. That's the pore, and carbon dioxide enters it for photosynthesis, and oxygen, which is a waste product of photosynthesis, leaves. This is also where water goes through. And then the chlorophyll, which is the green pigment of the thylakoid members of the chloroplast, right? And that's built within the thylakoid. So chlorophyll absorbs light energy, specifically blue, violet, and red. The light energy is used to boost the electrons to a higher energy level. Now you have accessory pigments that I'll talk about as well, but you can see here's our chlorophyll. We have that magnesium in the center. And so 2 electrons are going to leave because they have a higher energy. They've absorbed all this energy, right? Those are part of these photosystems. So these are going to have a chlorophyll A molecule in the reaction center, but they'll also have some accessory pigments, and they're going to be bringing all that energy to the chlorophyll A molecule in the reaction center. And that's built into that thylakoid membrane. So you have photosystem 2 and photosystem 1. They are just named from when they were discovered. So photosystem 1 was just discovered first. And they're getting a slightly different wavelength of red light You don't need to know, I don't believe, the specific wavelength. I didn't see that in the curriculum guide. All right, and they're going to be, however, connected by our electron transport chain. So what goes in? Well, water is going to go in, and light energy, which is absorbed by the chlorophyll, okay? And the water is going to actually be photolysed to replace the electrons that got excited, and that produces the byproduct of oxygen, right? Those electrons get excited, they get passed down the electron transport chain, they're going to create an electrical chemical gradient within the lumen, the inside of the thylakoid. That is going to be able to lead into the stroma using the ATP synthase. So very similar to what we saw with cell respiration. And then it goes to photosystem 1 in non-cyclic light reactions, where they get re-excited, the same electrons, and they're used by the enzyme NADP reductase to turn NAD plus into NADPH, also made on the side of the stroma. So it's made on the side of stroma because that's where it's going to be used in the Calvin cycle or Calvin-Benson cycle. So the formation of the ATP, the electrons go through electron transport chain. They're pumping hydrogen, but here's getting pumped into the thylakoid lumen. Electrical chemical gradient, high hydrogen here, low hydrogen in the stroma. This side feels relatively negative compared to this side. Lots of potential energy. Goes through the ATP synthase, and that turns ADP into ATP. Okay, so that's our formation of the ATP. Those electrons in non-cyclic are again replaced by the photolysis of water to make oxygen. So oxygen gas comes from water. Common misconception is people think it comes from carbon dioxide. But you may remember the experiment that they did with the heavy isotope of oxygen, using oxygen 18, so that they could trace where the oxygen came from. Cyclic is going to just make ATP, and that's because you need 12 NADPHs, but you need more ATP. So you need to make up the difference in ATP to run the Calvin cycle. So here, light hits photosystem 1, the electrons get excited, and they are going to go down the electron transport chain to make ATP, but then they're cycled back, right? Cyclic, right? Electrons are cycled back to that photosystem. So I am not going to make oxygen here because I don't need to replace my electrons, and I'm not going to have them go to NADP reductase because they're getting cycled back. Okay, so photosynthesis, those are our light reactions, but now that energy from light reactions, that ATP and NADPH, has to be used to run the Calvin cycle or Calvin-Benson cycle in the stroma, right? Because we want to fix this carbon dioxide. When they are used in that, those NADP plus and NADP will go back to light reactions to be reused. So now I'm doing the synthesis part. I'm going to do the making. Synthesis always means to make. So what goes in is carbon dioxide. It's actually 3 CO2, so you would double this to make a glucose, right? We're going to use the energy from our NADPH. So here it's 6 NADPH, for glucose would be 12. And we're going to use 9 ATP. So for a glucose molecule, it would be 18. Okay, remember I said there's a difference between the number of NADPH and the number of ATP. So the carbon dioxide gets picked up by that enzyme Rubisco, and it's going to be fixed using the energy from ATP and NADPH. So it's going to be reduced, and we're going to have an end product called glyceraldehyde 3-phosphate. You remember glyceraldehyde 3-phosphate as the end product of glycolysis. So that phosphate attached, so it has some energy attached to it. So the plant can use that to form glucose, 2 of those glyceraldehyde 3-phosphates, or because it's triose 3-carbon sugar and it has that energy, if it has to make something else from it, it could. Gives a little bit more flexibility. Again, this occurs in the stroma of the chloroplast of mesophyll cells for our C3, which are our most common plants, and also your CAM plants. If you remember, those are your plants that have to store their carbon dioxide at night as an acid and then wait for the daytime for ATP and NADPH to be made. and then it releases that carbon from the acid as carbon dioxide when the energy from the light reaction is available. They're trying to do that so they do not lose a lot of water, so they only have their stomata open at nights. All right. Rubisco, that is the important enzyme. It should pick up carbon dioxide and make G3P. But if there's a lot of oxygen, it can pick up the oxygen instead. It's the example that we use of the enzyme not being as substrate specific as it should be. Most enzymes are more substrate specific. So it picks it up and it runs it through and it uses up the energy if it has that oxygen picked up without making the G3P. Right? And so we have your three types. Most plants are C3. Rubisco is going to fix your carbon dioxide as normal. Your Calvin cycle is taking place in the mesophyll. So rice, wheat, soybeans. Sugar canes, for instance, is a C4. So here it's dealing with that photorespiration, Rubisco picking up oxygen instead of carbon dioxide. That's a problem for it. So instead, it takes the carbon dioxide, it fixes it, and it puts it in the bundle sheath cells. And so the Calvin cycle is going to occur in the bundle sheath cells that surround the vein of the plant. That makes sure that there's a high percent of CO2 and a low oxygen, so photorespiration is not a problem. It costs them energy. And then CAN plants, it's going to have the Calvin and carbon fixation occurring at different times. So the carbon comes in, it stores as an acid at night, so you don't have to worry about evapotranspiration occurring too much drying out the plant. And then that carbon has to be released during the day when NADPH and ATP are available. So that's cacti, pineapple, succulent plants. And again, this costs more energy than a C3 plant. So that's why we see most plants are C3 plants unless they have to they've had to adapt to either in C4 or photorespiration, or CAN plants being in a more arid area and losing too much water through evapotranspiration. This would be another example of a photosynthesis question. This is on DPIP. This is a publicly released exam, right? And this is the experiment and the data, right? And then the questions that they asked about it, right? So these are two questions that they asked about that data. So remember, I'm always telling you science process with questions. So this is the 2013 publicly released exam. I will most likely have you take that if we're not back in time, and I will give you an analysis sheet for that. It's not going to follow the curriculum and exam description exactly, but at least it'll give you some feedback. All right, so comparing respiration, we're going to see in plants and animals. We're also going to see some form of respiration, anything that's living. It needs oxygen, and in food, it produces carbon dioxide, water, ATP, and NADH, right? If it's aerobic, it's going to occur in the mitochondrial membrane and matrix. Anaerobic is going to have that oxidative phosphorylation with the proton gradient. Photosynthesis, this is in plants. It's also in things like algae and about 1/3 of Euglena phytoproteus as well. And it's going to need carbon dioxide, water, and sunlight. It produces glucose, oxygen, ATP, and NADPH. It's going to occur in the chloroplast, thylakoid, and stroma. It has photorespiration as a problem because Rubisco. But again, we see a proton gradient across the membrane. So it's important that you see that chemoosmosis to generate ATP is in both cell respiration and photosynthesis, right? And just please don't get your NADH and NADPH confused, all right? And make sure you know your equations. These are not a fully balanced equation. So for instance, over here should be 12 water, and we should have an addition of 6 water over here, right? This is a simplified one. So fitness, the last part of Unit 3. This is to explain how variation, number, and types of molecules within a cell affects the ability of the organisms to survive and or reproduce in different environments, right? So just taking directly from the curriculum exam description allows organisms to respond to a variety of environmental stimuli, provides organisms a greater ability to survive and or reproduce in different environments. We kind of covered this throughout the year, and with some emphasis, obviously, during the evolution unit. So with our phospholipid bilayer, they can change to adapt to an environmental temperature. So cold, we're going to see an increase of unsaturated phospholipids. So some ones with the double and triple bonds, so that they don't stay together and keeps it more fluid. If it's warm, you want more saturated ones so that it's more viscous. And cholesterol, I apologize, that helps again with temperature change, right? So that's in between these fatty acids. Hemoglobin, that's going to change based on developmental stage. So you have your embryonic one, that's demonstrated here in pink. You have your, also you have here the fetal hemoglobin, and you can see that is going to be, has a higher affinity, I don't know if it shows it here, but it has a higher affinity for oxygen. Then you have your adult hemoglobin. So a fetus needs more oxygen for cell respiration because it's undergoing a lot of growth. You're having more mitosis. So you need more ATP to run that. So it has to compete with its mother for oxygen. So by having that fetal hemoglobin, having a higher affinity, right, it can pick up more oxygen here, okay, than the adult hemoglobin, right? And so the fetus can get the oxygen it needs for cell respiration and to have therefore the ATP, the energy available for growth. And then accessory pigments and chloroplasts. This is another example. This allows plants to absorb more wavelengths of light for photosynthesis. So for instance, if you look at a forest, the leaf canopy is going to get a lot of that green light. Some will still get to the plants on the bottom, but they're going to need some accessory pigments to absorb other wavelengths of light too. Okay. And so you would see, if you look at, I think Coleus that comes out soon, a lot of them will have like red color to them or some purple tinge to them as well. And that's because they're using some accessory pigments. Okay. So I hope that you found that useful, and feel free to email me any questions that you may have. And I will try to either address them with you directly, or if I have a lot of them, I will make another lecture to try to address them. Or if we can, we'll try to do a Google Hangout or chat. Okay? Thank you, and have a good day.